Hopefully I didn't mess up too many people by showing up on time today. That would be unfortunate. Yeah, they tell me that even though we're giving a take-home final, I'm required to come and pretend to give class when the final's scheduled just because, like, strictly hypothetically, there could be classes on campus with lots of football players in them where they didn't give a real final and nobody showed up for anything. Not this would ever happen in our illustrious institution of higher learning, but if attendance thus far is any indication, I'm thinking maybe I won't bother, but anyway, I should lecture and not ramble. So today we're continuing on the discussion of just kind of random topics that are useful in bench science but don't really fit into a standard statistics curriculum. And today we're covering cluster analysis, as you can see here. It's particularly if you ever work with gene expression data at all, this is one of the most commonly used methods out there. And I thought it would be worthwhile to at least give an overview of what it's all about. So, cluster analysis, what does that mean? The idea behind cluster analysis is that you want to try to group a data set into subsets or clusters such that these subgroups are relatively homogeneous. The observation in a given cluster are more similar in a certain sense than observations in different clusters. And in some cases you want to form a cluster hierarchy that means within each level of the hierarchy, clusters in the same group are more similar than those in different groups. So, simple examples, if you had data that looked something like this, that you might see that there's kind of a clump of points in this corner, a clump of points up here, a clump of points down at the bottom, and you want to partition the data set into these three different clumps and say, okay, this is one cluster, this is one cluster, this is one cluster, and so on. Why would you want to do this? Well, one common example that's resulted in a long list of high-impact publications in recent years is if you have gene expression data from cancer patients and you want to try to find subtypes among these patients. For example, it's well known that not all tumors will respond the same way to treatment, that some are more likely to respond than others, or some may respond more effectively to certain types of treatment than to other types of treatment, or some just may be more aggressive. A common example of this, a lot of work's been done, is breast cancer, that right now, when a breast tumor is detected, the standard treatment is surgically remove the tumor, and if you think there's a high risk of metastasis, then you can do chemotherapy as well. Now, chemotherapy is not fun. It's something that you would want to try to avoid if at all possible, but most doctors tend to think along the lines that if you get chemotherapy and don't need it, you lose all your hair and get really sick, but you're alive. Whereas if you don't get chemotherapy and you do need it, you might die. So most doctors tend to err on the side of doing the chemotherapy. There's been a fair amount of work in recent years on trying to find subtypes of breast cancer based on the gene expression profile of the tumors to try to predict more accurately which tumors are more likely to metastasize so that you can provide the more aggressive treatment to the more dangerous types of tumors. Or another common example of cluster analysis in the bench science world is if you have a set of DNA sequence data on several different species and you want to figure out how they're related to one another. That been a while since I've studied this, but my recollection is like the sequence of hemoglobin or the sequence of cytochrome C are two of the favorite places to look at this. Say you have a bunch of insects, a bunch of bacteria, whatever the case may be. You have a bunch of sequence data. You want to say, okay, these two species have very closely related sequence data. They're probably close cousins of one another, whereas these are separated more distantly, type of thing. Or another possibility is if you want to find patients with similar characteristics that are developing a complex disease. I work in chronic pain, and I'm writing a paper on this right now. The idea is that diseases like temporomandibular disorder, fibromyalgia, can be caused by a number of different things, and there may be some patients whose disease is caused by just trauma you get in an accident. Another set of patients there may be caused by central problems, centrally mediated things in the central nervous system. You can tell I'm a real expert on this, but the idea is that if somebody has pain in their jaw just because they got hit in the face with a car accident, well, then maybe you just put them on painkillers and tell them to go home because it'll get better on its own, whereas someone whose pain is driven by more central neurological processes may require more intense treatment. Well, these are all examples of when you may want to try to take a data set, break it into homogeneous subgroups, in hopes that it will tell you something about the underlying biology. Oh, to do cluster analysis, the first thing you need to do is define a dissimilarity measure that, as I said, the purpose of cluster analysis, you want to group your data into quote-unquote similar objects. In order to do that, you have to define what you mean by similar. And the way that's normally done is you define a dissimilarity measure between each possible pair of observations, sometimes called the distance measure between observations. You can say these two observations are similar to one another, or these two are more distant. And the most commonly used method, and the only method I'm going to discuss at all today, is the simple Euclidean distance, which hopefully you remember from algebra class. If you want to find the distance between 2 points, you can use the Pythagorean theorem and say it'll be the square root of a squared plus b squared plus c squared, and that will easily generalize to higher dimensions. And so essentially, if you want to find the quote-unquote distance between 2 points, you just take the x-coordinates, subtract from one another, square it, the y-coordinates, subtract from one another, square it. Repeat that for all the other coordinates, add it together, and you define that to be your Euclidean distance. And there's the mathematical formulation for the Euclidean distance. If you have x1, x2 up to xp, then your Euclidean distance will be x1 sub p minus x2 sub p squared. Sum that over all the possible values of p. And the intuition is just the square of the distance between the 2 points if you measured it using a ruler according to the Pythagorean theorem. There's some other distance metrics that are possible, but we won't discuss those here. And one of the most common, perhaps the most common, well, I don't know, nowadays, thanks to microarrays, hierarchical clustering may be more popular, but definitely one of the two most common cluster analysis methods is what's called k-means clustering, which functions basically as follows, and it looks like the numbering of my steps got lost when I switched to Keynote, but oh well. That the first step, you just randomly assign each observation to one of a set of possible clusters, and within each cluster you find the means of the observations, and then you take the distance between each observation, each of the cluster centers, and assign the observation to the closest cluster. So then you have a new set of clusters, and you find the mean of the new sets of clusters, assign each point to the closest cluster, keep repeating this process until the process stops and the cluster assignments don't change from step to step. This next slide illustrates how this procedure works. In this case, rather than randomly assigning each observation to initial cluster, it just randomly chooses 3 points to be the initial cluster centers. That works too. In this case, it says let's take these 3 points to be our quote-unquote cluster centers. And then it calculates the distance between every other point and those 3 points. It says the closest observation to the red dot at the top is that one right there. It gets assigned to the red cluster. All these observations down in this corner are closest to the green dot here in the center, so they all get assigned to the green cluster. Similarly, all the points on this side are closest to the blue dot, they get assigned to the blue cluster. So then we end up with a new set of clusters, the red cluster, the green cluster, and the blue cluster. So then this process repeats. You calculate the mean of the red cluster, which is probably around there. You calculate the mean of the green cluster, probably somewhere in there. Mean of the blue cluster, somewhere in that area. And then now this point here is closer to the red cluster than the green cluster. It gets moved up into the red cluster, I think, as do these points down here as well. Ones over here don't change, so you end up with the red cluster up in this corner, and it looks like a couple of the dots at the top disappeared. A green cluster down there, a blue cluster up there. If you took another step, I think there's a good chance these 2 points down here would end up in the green cluster. Those two would stay the same, and then you'd be done, something like that. In practice, the details don't matter that much because, as usual, R will do this for us. And choice of initial clusters, in general, it's random, but unfortunately, the results of the k-means procedure can vary quite a bit depending on the choice of initial clusters. So usually when you do k-means clustering, what you'll want to do is run the procedure several times with a bunch of different starting points and then choose the quote-unquote best set of clusters across all these starting points. Usually best means that the clusters are quote-unquote as compact as possible. That is, the distance between a given point, the sum of the distances between all the points in their cluster centers is as small as possible. In my experience, if you choose 10 different initial starts, that's usually more than sufficient. Oh, an example of cluster analysis. This is using a data set that's built into R on earthquakes that basically scientists recorded the latitude and longitude of the epicenter of 1,000 earthquakes that occurred off the coast of Fiji. So one might want to ask, is there any sort of pattern to where these earthquakes occur? Could it be that one set of earthquakes occur along one fault line, another set of earthquakes occur on another fault line, and these fault lines are the locations where earthquakes are most likely to occur in the future? So if you plot this data in R, the data looks something like this. And I mean, just eyeballing, it looks like there's probably a cluster of earthquakes up here, another cluster of earthquakes down here. So the question then becomes, can we use R to detect these clusters and be able to say more rigorously that this is a homogeneous set of earthquakes and this is a homogeneous set of earthquakes? Oh, to do this in R, the function is called k-means. Like I said, this data set is built into R. It's called quakes. I just take the first 2 columns of the quakes data set that are the latitude and longitude. Then I say, let's do k-means clustering on the latitude and longitude of this data. the second argument here is the number of clusters, k. In this case, we want 2. And then I say we'll take 10 initial starting positions. So, in general, if you say k means with the data set x, number of clusters c, number of starts, that will cluster your data x into nc clusters using k-means clustering. And this parameter n starts equals 10 will tell R to try 10 different random starting positions. And my personal recommendation is that anytime you use k-means clustering, you should always include n starts equals 10. The default is 1, but you'll almost always get better results if you use 10 different random starts rather than just 1. And this k-means command will return a list with 4 components. There'll be a component called centers that are just the cluster means of the cluster means of the 2 clusters. So in this case, there's one cluster with a center at about minus 21.9 degrees latitude or 21.9 degrees south latitude and about 182.4 degrees longitude. A second center that's at about minus 15.9 and 168.3. And the second output is size that gives the number of observations in each cluster. One cluster has 795 observations, the other has 205. Then within sum of squares gives the within cluster sum of squares for the 2 clusters, and then $cluster just gives the cluster assignments of each observation. That tells you that the first few observations are all in cluster 1, then one's in cluster 2, and then you see more in cluster 1. There's a lot more in cluster 1 than cluster 2 here, but some end up in cluster 2. I only printed the first 100. There's 1,000 observations in this data set, and it would fill up the whole page. So, yeah, I think I just went over most of this. It'll give you the center, the number of observations in each, the within cluster sum of squares, and the cluster to which each observation was assigned. And here I plotted the earthquake data color-coded by cluster, and it looks like what k-means found was consistent with what we got from eyeballing the data. There's one cluster of earthquakes down here colored red, another cluster up here coded in black, suggesting that indeed there may be 2 separate fault lines leading to 2 different sets of earthquakes. So, how do you choose the number of clusters? If you've been paying attention, you notice that to do cluster analysis, we have to specify the number of clusters, and typically in advance, that's not something that we will know. And you can't just choose the set of clusters that minimizes the within cluster sum of squares, because it's fairly easy to show the within cluster sum of squares will always decrease as the number of clusters increases. Determining the optimal number of clusters is a tricky problem and often quite subjective, but there's some rules of thumb that you can follow. The folklore is sort of like when you do principal components analysis, that what you can do is you can try k-means clustering for a bunch of different values of k, and record the within cluster sum of squares for each possible value of k, and then plot the within cluster sum of squares versus k, and then just look for an elbow in the graph. The idea is that if you see an elbow in the graph, that means that the within cluster sum of squares decreases drastically when you go to that number of clusters, but when you go past that number of clusters, the improvement's minimal, suggesting that where the elbow is where you should saw it. Maybe a picture will make this clear. That here's the commands in R for how you can plot this graph. I know this is a little bit convoluted and you don't need to worry about the details here, but if you ever need to do some practice, you can just copy this code. This most likely will not be on, well, definitely won't be on a homework assignment, and it's unlikely that I would put this on the final, so don't stress over it too much, but if you ever need it, there's the code. And basically, this graph shows the log of the within cluster sum of squares versus the number of clusters. You can see when we do k equals 1, i.e. no clusters at all, the within cluster sum of squares is quite large. There's a pretty drastic decrease when we go to 2 clusters. Once we go past 2, the additional improvement is quite gradual. So if you see a graph that looks like this, you say, okay, it looks like k equals 2 is the appropriate number of clusters. You get a big improvement from 1 to 2, small improvement after that. 2 is probably the correct cutoff. That's the quote-unquote elbow that I was talking about. And this is obviously quite highly subjective. Two different people could look at the same graph and draw different conclusions about the number of clusters needed. And figuring out the correct number of clusters is actually a reasonably active research area. And the various methods are well beyond the scope of this class. If you're doing a paper and you need to be really rigorous about the number of clusters you chose, you're welcome to shoot me an email or your other favorite statistician an email if you need help with this. And so that's the brief introduction to k-means clustering. Now we're going to consider hierarchical clustering, which is viewed as a bottom-up algorithm. Basically, the idea is that k-means is sometimes referred to as a top-down clustering because it takes the full data, splits the full data in two. Bottom-up clustering works in the other direction, that you start with individual observations, find the observations that are similar to one another, group those together, and then gradually work your way up to the full data set. Oh, hierarchical clustering and my numbering got lost again, but the way hierarchical clustering works is you look at all possible pairs of observations in your data set, find the two observations that are most similar to one another, cluster those two observations together. And once they're clustered together, you combine them in some way or another. The simplest way to quote-unquote combine them is you just average those two together and let their average coordinate be the coordinate of that cluster. Then just repeat this process. Look at every possible pair of observations again and combine the two most similar observations, including the two observations that were combined together in the first step, and then you just repeat this process until all of your data has been combined into a single cluster. This data set here illustrates how hierarchical clustering works. Let's say you have a data set that looks something like this. So, the way hierarchical clustering works is it looks at every possible pair of points in this data set and finds the 2 points that are most similar. And this particular data is pretty much a push between B and C and D and E. So the first 2 steps, B and C, get combined together to a B-C cluster. D and E get combined together to a D cluster. So now we're basically down to 4 points. This point here, those two combine, and that one. Oh, the next step, F is close to the DE cluster, so it gets combined into a DEF cluster. So now we have cluster A, cluster BC, cluster DEF. And these two clusters down here are closer together than A is, so it gets combined into a BC-DEF cluster. The very final step, A gets combined to the BC DEF cluster, and now it's all one big cluster, and the procedure stops. So you end up with a cluster hierarchy that looks something like this graph here. And there's a number of different ways to define similar observations. And the simplest that I just described is the lowest Euclidean distance. Sometimes in the microarray world, they will choose the highest correlation or things like that. And similarly, there are several ways that you can define the distance between an observation and a cluster. The distance between observation, you can take the distance between observation, the average of the points in the cluster, the distance between the observation and the furthest or the closest, or whatever the case may be. Usually it doesn't make a huge difference, but you can try several different variants if you're trying to find clusters that work especially nicely. And one common example of cluster analysis is, or hierarchical clustering in particular, is if you work with microarrays at all. People love their hierarchical clustering in microarrays. That I think the main reason for it is because it draws cool pictures. I mean, it's kind of silly that that's what drives science, but you know, whatever. This is output of hierarchical clustering in a paper I worked on back in my grad school days. It's a data set on acute myeloid leukemia, and it's been shown in the past that the prognosis of leukemia, the prognosis of acute myeloid leukemia depends on what type of karyotypic abnormalities you see in the tumor. That if, I'm trying to remember that, like, if there's an inversion on chromosome 16, that's associated with poor prognosis, or if you see a transposition between chromosomes 8 and 21, that's associated with good prognosis. I can't remember anymore, but it's something like that. But one way or another, in this paper we showed that if you do the hierarchical clustering on a microarray data set from AML patients, see that depending on the karyotypic abnormalities in the tumor, their gene expression pattern is different, which was kind of reassuring to see. That, and this was just hierarchical clustering, they just found observations that were similar, clumped them together, and kept on working their way outward with respect to both genes and patients. Incidentally, in this case, they did the clustering on both genes and observations, which is why you see the little tree diagram on both the left side and the top. Oh, an example of hierarchical clustering in R. This is similar to what you would do if you're trying to figure out which species are related to one another using their DNA sequences, but unfortunately the data set built into R doesn't have DNA data. It just has a list of 28 animals, and it has the weight of their bodies and the weight of their brains. Let's see if we can try to group animals into similar species of animals just using these brain weights and body weights. So, to do this in R, you can use the commands dist and hclust. Dist calculates the Euclidean distance between pairs of observations in a data set. Given those pairs of Euclidean distances, the hclust command does hierarchical clustering on those distances, and then the plot command says to plot the cool tree diagram for the resulting hierarchical clustering. So what does this look like when we run it? I guess I do a little commentary on this. If you say hclust dist, that'll do hierarchical clustering on X. dist of x will calculate the pairwise Euclidean distance. If you wanted, you can define some other distance metric and use that instead. So we do this on the animal's data in R, and we see that one animal jumps out as a clear outlier and does not cluster closely with anything else. It is the Brachiosaurus. Well, okay, that's somewhat reassuring. If we're going off of brain sizes and body sizes, we should expect the Brachiosaurus to be a huge outlier. The next, you see down at the bottom, the Triceratops and the Diplodocus clustered together out here, separate from any other animals, but still a ways away from the Brachiosaurus. Okay, fine. Two animals closest to the dinosaurs are the Asian elephant and the African elephant. So all that's fairly reassuring. And if you look down at the bottom, you see kind of what you would expect to see similar animals clustering together, that you have like the cow, the horse, and the giraffe all together down at the bottom. Similarly, you see like a mouse. I can't even point at the right place, but see like mouse and rat also clustered very closely together at one of the bottom levels. Rabbit, mountain beaver, sort of thing. I mean, not all of these make perfect sense, but by and large, given that we only have two rough measures of brain size and body size, this doesn't do a terrible job of clumping these animals together. There's a lot of different flavors of hierarchical clustering out there. I mean, all I can do for this class is scratch the surface. If you're not sure which version of hierarchical clustering to use, then your best bet is just to talk to somebody who's experienced with the methodology. I'm happy to talk to people outside of class, or just find some other statistician who's worked with this previously. So, when should you use k-means and when should you use hierarchical clustering? Well, the folklore is that k-means does a better job of identifying quote-unquote gross features in the data, whereas hierarchical clustering is better at identifying fine features. That if you just want to split your data into two subtypes at the top, supposedly k-means is better for that, whereas hierarchical clustering, if you want to be able to find a group of observations that are really similar to one another, but you don't necessarily care if your top level clusters are correct, hierarchical clustering is better. Now, if you ask me to point to the paper where somebody showed that, I've never seen it. I'm not sure if it exists, but my dissertation advisor in grad school was one of the gurus of cluster analysis, and that's what he told me, so I just assumed that he knew what he was talking about when he said that. And another reason the people like hierarchical clustering, probably the single biggest reason that people like hierarchical clustering is, to be honest, you can do the pretty graphs, particularly of your microarray data. I mean, this... the hierarchical clustering program that Michael Eisen wrote for microarrays, I don't think I've ever seen a single microarray paper that didn't use that program. The last I checked, that paper had something like 10,000 citations. It's like, do hierarchical clustering that's been around for 30 years and draw cool pictures of it? Seriously? Like, why didn't I think of that? And so my paper would get 10,000 citations, but, you know, oh well, I guess that's life. So, things to remember for today, and wow, I guess I went through today's lecture pretty quickly, is that you can group a data set into similar subsets using cluster analysis. The two most commonly used approaches are k-means clustering and hierarchical clustering, and there's advantages and drawbacks, and if you want to do something more sophisticated with your cluster analysis, then just draw pretty pictures. It's a good idea to talk to somebody who has experience. They can help you do it correctly. And the R commands to do k-means clustering and hierarchical clustering are here. Any questions about any of today's material? I will see, hopefully, at least somebody on Monday then. Like I say, please don't hesitate to email me if there's anything that you want to learn about that we're not covering, because, yeah, particularly when attendance is sparse, if you want your personal lecture in statistics, it's a good chance to get it.